Record it. That's all right. Okay, that's it. And Lead is going to introduce our guest speaker today. So.
Hi.
Please.
Okay, we need the before one.
Yeah.
Hi, everyone. Thank you so much for being with us today. And it's a great pleasure to welcome Heather Stapleton, and well-distinguished environmental health scientist with long-standing expertise on exposure assessment with different techniques, targeted and untargeted, and professor at Duke University. She's gonna tell us about a lot of novel stuff and also stuff that perhaps you are familiar with it. Please save some questions for the end so that we can have a fruitful discussion. Thank you.
Thank you. All right. Are we all good to go on the, all right, thanks. All right, so, well, good morning, almost good afternoon, everyone. Certainly want to thank you for inviting me to be here today to talk to you about some of our research. So yeah, I'm Heather Stapleton. I'm at Duke University, and I'm an environmental chemist by training, but I like to refer to myself as an exposure scientist. And I'm really interested in this concept of the exposome, which I'm sure many of you have heard about. Right, it's very dynamic, multifaceted. The exposome seeks to assess the totality of exposures over the life course and understand how these stressors impact our health. Right, so I think about this a lot. How are we going to do this? This is a very daunting challenge. And and today a lot of the studies that we're doing, particularly in epidemiology, they're case control, they're cross-sectional. We're looking at exposures at one point in time, right? When I think about this, I think about this part over the life course. We need to understand exposures over time. And that's very hard to do in some of these studies. And I understand we're resource limited. That's why a lot of our studies are case control and cross-sectional. There are prospective studies, certainly, but many of those are once a year or once every 2 years, unless it's a pregnancy study. So it's hard to get a lot of that data. And I think that's very important. And I think this is where wearables and wristbands can help fill some of this data gap in trying to understand what these exposures that are occurring over time. And hopefully help us understand how our behaviors, our activities drive exposure, housing characteristics, consumer products, our occupations, how these relate to temporal changes that occur. Because I think this is really important to understanding the exposome. Right. So I want to first say I am not the first person to introduce wristbands or to use them. These are actually introduced by Kim Anderson and her PhD student, Stephen O'Connell, in this paper in 2014. But I remember when this was published and just thinking this is a great idea. I at that point am doing a lot of work in indoor environments, looking at children's exposures and looking using dust, and we were using hand wipes. And I said, oh, this is a great idea. We should use this. And we partnered with Kim to do some kind of seminal work to understand what are the strengths and limitations of the wristbands. Right. So a little bit about what we know. There's still some things we don't know. But the data collected to date suggests that the exposure routes best captured by the wristband include inhalation and dermal exposure. And I would argue that they potentially capture exposures that are occurring, particularly in kids, through inadvertent dust ingestion. And I'll come back to that. I think this is still being debated in the field, but I feel like there's data to support this. Now, the exceptions are that the wristbands are obviously not going to capture dietary exposures, right? What we're eating, what we're drinking. That can be a strength. That could be a limitation, depending on your question. And they're not so good for use in metals. A lot of what my group does focuses on organic contaminants, a lot of persistent organic contaminants, right? So metals don't seem to stick or absorb to this very well, although there are groups trying to modify wristbands to enhance their capacity to detect metals, and particularly lead, because it's important in a lot of areas. Right, so we buy wristbands from a commercial vendor. They're actually pretty cheap, 10 to 15 cents a piece, depending on how you customize them. After they're clean, you keep them airtight, and they serve as basically a passive sampler while you're wearing them. And the thought is that chemicals will accumulate. Do I have a laser pointer at all by chance? Okay. Right, they'll accumulate over time in a linear fashion, eventually reaching this equilibrium steady state condition. But typically we're trying to use these in a way that we're in that linear uptake phase. Most of our studies, people are wearing these for somewhere between 5 to 9 days continuously. And then the slope of that line is going to be a function of the concentration gradient between the wristband and the ambient environment and the phys-chem properties. Right, but what's nice about these is that they're capturing our exposure in all these multiple microenvironments where we spend our time, right? Particularly the home environment, we spend a lot of time there, but also commuting, our work environment, And if our question relates to how do exposures are associated with health outcomes, you want to be able to account for your exposures in all these different microenvironments. However, you might separately have a question where you're just interested in exposures, maybe in one compartment, like the occupational setting. We have some work in that case where you just then have the participants wear it in an occupational setting, right? So it's a little bit flexible in terms of your question. All right, so what I wanted to do today for the rest of the seminar is talk a little bit about, strengths and limitations of the wristbands, what we know. These are some of the questions that we receive most commonly. How well do they correlate with internal dose? How do they compare to a spot urine sample? How variable are they around the wristband? Does showering wash these off? So I'm going to talk about some of the data we've collected since we started working with these to try to answer some of these questions. And then I'm just going to dive in a little bit deeper on two of my favorite projects. One is with working with firefighters using wristbands, and another one is a One Health study working with dogs. And then I'll briefly mention this other potential use in toxicology. All right, so I'm going to first start just by talking a little bit about our methods. All right, this is a photo of what we call a wristband kit. We prepare these in my laboratory. Right, there is a Mylar bag there that's resealable on the top. It has a sticker on it, so you can have the participant's ID number on there. There's a date, a location for date and time on and date and time off. Right, this is a time-dependent accumulation, so it's very important to record those details. Inside the Mylar bag is this aluminum tin. Right, you can just, it's a screw top. There's a top to it, so it keeps it airtight, and the wristband is inside. So when we have people wear them, we get them returned in these Mylar bags. We keep the tin typically in the freezer, and then when we analyze the wristbands, we're actually only taking a small piece of this because there's so much that accumulates on this in this wristband. We don't need that much to be detectable by mass spectrometry. So actually the piece that we're analyzing is only about a fifth of the wristband. It's about 0.7 grams. The rest we archive for other analyses, so they go back into the freezer. This is a little bit of overview of what goes on in our lab, just quickly. The top panel there talks about our pre-treatment. So when we buy these from commercial producers, we bring them into the lab. In the past, we've done a lot of solvent cleaning soxhlet extraction to remove the background contaminants on there. I didn't really like using a lot of solvent, so we're now moving to this system where basically we're heat cleaning them. So we heat them up to 250 degrees under vacuum with nitrogen purging. And that actually helps us achieve that same level of cleanness in the wristbands compared to the soxhlet extraction. So that's what we're doing now. We deploy these, we get them back in the laboratory. Kim Anderson's group has done a study that has shown that most chemicals are pretty stable on the wristbands for up to 6 months at room temperature and even up to 35 degrees Celsius. But we encourage people to get them into the freezer when they can. It's just good and kind of reduces the risk that things could be volatilizing and off-gassing from the wristbands over time. In the laboratory, as I said, we take very small pieces of these. Basically, we cut them up into really small pieces and we put them in a test tube with organic solvents and sonicate them to extract them off the wristbands. We do have a very quick dispersive fluorocell cleanup that helps remove some of those skin oils in particular that can interfere with our analysis. And then that extract we take and we inject it onto 3 different mass spectrometer systems that are designed and are sensitive to different classes of compound. Our primary panel, the workhorse, is all the way to the left where it says EI at the end. These are all thermo Orbitrap high resolution mass spectrometers. The EI helps us collect data for targeted analysis of, you can see the list on the bottom, brominated flame retardants, organophosphate ester, flame retardants, plasticizers, pHs, pesticides, PCBs, and several industrial chemicals and personal care products. Then we have a second system, an exactive that we use for high molecular weight brominated flame retardants, because we need to use negative chemical ionization mode to be more sensitive. We have a short column with a thin film to be more sensitive. So we look for several of these high molecular weight brominated flame retardants and some dioxins, furans at the same time. And we're actually trying to optimize that method to collect information on chlorinated paraffins right now as well. And then we have a third system we use that's in positive chemical ionization mode for the volatile PFAS. Everyone's really interested in PFAS these days. Most people are using LCMS, focusing on the non-volatile PFAS, but there are, particularly in indoor environments, there are several classes of PFAS that are being used in building materials. And so we want to have the ability to look for these volatile PFAS as well. So we have a panel on that instrument. And then secondly, we do also look for some of these non-volatile, more polar compounds. But typically to do that, we'll do a separate piece of wristband with a methanol extraction, and we run those on our LC tandem mass spec. It's an Agilent 6460. where we do have a whole panel of PFAS that we look for, including the legacy PFAAs and some of these other precursors. And we also have a panel for looking at some of these chemicals and personal products like parabens and triclosan. Right, and you also see a snapshot down there that says suspect screening. So that first panel, when we collect data, we're collecting data on in a targeted fashion because we want to be able to produce results using calibration curves and be accurate in our estimates. So we're producing data in parts per billion typically, but we run it in a way that we collect all the other data that's there. What else is in the sample? This is a non-targeted analysis. And you can see on the right, the top chromatogram is an example of a chromatogram from a wristband sample extract that we ran in the EI panel. Up on the right, you can see it says 1,617 molecular features found. So those are the individual signals or peaks that were resolved from that chromatogram. Theoretically, then 1,617 different chemicals on there. And you can see very little to nothing in the blanks, and we blank subtract these. So based on my experience and my assessment of the data we collected so far, we see these really unique patterns, right? Or what I call chemical fingerprints by using the wristbands. So it kind of gives us the ability to look at these different chemical mixtures that different people are exposed to, almost like a chemical fingerprint, right? Which I think is important for helping us to understand this personal exposome theory. When we do suspect screening, we actually collect the data at the same time. We collect the data for the targeted analysis. And this whole method has been being spearheaded by Nicholas Herkert, who's a research scientist in my group. So he's helped develop this whole workflow. We use the thermo deconvolution program on the mass spectrometer to kind of do our peak picking and our peak alignment. It pulls out those features, and then we compare them to 3 different mass spectral databases. One we've created in-house. One is basically a pesticide library from Thermo. And then we have access to the NIST-23 mass spectral library as well. And then Nick has developed kind of a unique algorithm that weights the tentative identifications in a certain way based on some metadata to kind of produce our list of what those features likely are. And we do a lot of visualizations with volcano plots. And so I'll show you and give you some examples of those when we get to the other studies. So let's focus on some of these questions that we get a lot first. Are measurements of chemicals on the wristbands correlated with internal dose? I think that was the first project we did, working with Kim Anderson's group, because we thought this was very important to address. So here I'm just giving you examples from 3 different publications, looking at the correlation between what accumulated on the wristband over the time period in which it was worn, relative to the established biomarker of exposure. In this case, all 3 of these are measured in urine. So what we typically do is have participants wear these, usually for 5 days in these cases. And then we ask the participant to collect a first morning void urine sample on days 1, 3, and 5, and we pool them. And so the y-axis there is showing you the measurement of the established biomarker in the urine from that pooled urine sample. So on the left, you can see the data and the nice correlation for this chlorinated organophosphate flame retardant that goes by this acronym TDCP. In the middle is benzoyl phthalate. So this is a phthalate that's really commonly used in things like vinyl flooring. And then the last one here is the pesticide DEET, right? So that's used as a mosquito repellent and things like Deep Woods Off. What's interesting about this DEET, you can see that's a very nice correlation, and that actually came from our suspect screening. That didn't come from our targeted data. So what's plotted on the x-axis here is actually area response and not nanograms per gram. So the rest were in our targeted panel. DEET actually came out from our suspect screening, and you can see a very nice correlation with the urinary metabolite. DEET is now in our target panel, but at the time we did this study, it was not, but was clearly identified through the suspect screening. We've also done some comparisons with measurements in blood. So here are 2 brominated flame retardants. The one on the left is BDE47. This chemical in particular has a very long half-life in the human body. It's estimated that its half-life is about 1.8 years. So obviously, if you're measuring exposure over 1 week, you're only looking at a small snapshot of exposure relative to the lifetime of that chemical in the blood. But we still saw a statistically significant correlation between the measurement in the blood and the measurement on the wristband, likely because most of these participants had lived in the same home for the last couple of years. And previous studies had clearly shown there was a relationship between levels in dust and levels in the blood for PBDEs. So we think the wristbands are picking this up as well. And then on the right is a different brominated flame retardant, goes by the name of DECA BDE or BDE209. We had a smaller number of participants in this study, but we do see a statistically significant correlation This chemical, in contrast, has a shorter half-life, usually about 2 weeks. But one of the reasons I said that wristbands are useful for looking at exposure routes that might occur through inadvertent dust ingestion is because of that data right there. There's been a number of studies looking at this flame retardant in particular, and the consensus is that most people are exposed, particularly children, through inadvertent dust ingestion. And this is actually data from our children's cohort. So these are all toddlers between the ages of 3 and 6. So I think what we're seeing is that we do get dust that's kind of kicked up, it accumulates and reflects likely what some of these kids are exposed to. Now we get asked about PFAS a lot. Can you use wristbands to measure PFAS exposure? The answer is very complicated and depends on your question, right? Most people are interested in the legacy PFAAs like PFAS and PFOA, which you see there on the left. Most of our exposure to those two compounds is through our diet, drinking water, food packaging, levels in our food supply. We do look for them in wristbands. We actually see them, particularly in our firefighter study, but levels are very low because these are not volatile. They're not going to be present in the air. When we see them, the levels are usually less than a parts per billion, around 0.1 parts per billion. But what we do see on wristbands are these precursors, particularly the volatile precursors, right? So something like the fluorotomer alcohols or these fluorotomer phosphate compounds. These are chemicals that we know are used in things like paints, carpeting, stain repellent treatments. So we do pick these up and more frequently and more abundantly on the wristband. It's a little more challenging to understand what this means for exposure relative to PFAS in our body because we don't know as much. These are not persistent like the legacy PFAAs. They are metabolized, they are degraded. Now they have the potential to degrade into the PFAAs, but we don't know over what time scale in the human body. There are some data suggesting some of these are conjugated in the body and could be excreted in the urine. But we don't have good biomarkers of exposure for these precursors because right now there's still the assumption that they're going to go down to these PFAAs. I think this is a big data gap and one I'm hoping we can help answer with some of the wristband. But what I like about them is that they're capturing clearly exposure to these precursors in the indoor environment. And again, depending on your question, that could be very useful information. Now, we do have 2 studies right now in which we are collecting paired samples of wristband, blood, indoor air, and dust for PFAS specifically, one here in California and one in Michigan. So we do have data from Michigan, and I'm going to show you some of the, our preliminary data. I know my student presented this at SCEHSC last fall. Here you can see we did see a very nice correlation. This is from our Michigan cohort between methylphoce on the wristband and methylphosa in the blood, right? Correlation coefficient of 0.9. So you can see the concentrations on the wristband on the x-axis range from about 0.1 to 20 parts per billion. Methylphosa is one of those 7 PFAS that are a focus of the National Academy's medical guidance recommendations. So you look at the sum of those. It's not as abundant as PFOA and PFAS in blood, but it is commonly detected. And I think our data clearly suggests that this is probably the one PFAS where the exposure is coming from the indoor environment and not from drinking water and food. And we have now replicated this in a second cohort. We see the exact same trend, right? And this biologically does make sense. If you look at the chemical structure of methylphoce and look at that terminal sulfonamide group, it has an alcohol, methylphoce. That alcohol is oxidized to a carboxylic acid, which is what you see in methylphosa. And that is often a first transformation step that occurs. Now, whether this occurs outside the body or inside the body, I think that's a separate question we don't know the answer to. All right, but I showed you many examples of chemicals that correlated. Not everything is correlated, right? So I want to make sure we talk about ones that are not. We see just as many not correlated as we do see correlated. And I think this just depends on what the major exposure source is for these compounds. So for example, here's one of our studies with chlorpyrifos. This is in our panel. We do detect it pretty commonly in some of these cohorts. Here's an example. This is from one of our studies, had 30 participants. We detected chlorpyrifos in the wristbands in 83% of the samples and 100% of urine. If you look at the correlation, you can see they're not correlated. These are also pooled urine samples. And I think this makes sense because the data does suggest, or research suggests, that our exposure to chlorpyrifos is from food, right? There's residual pesticide levels on some of our food. So I think what we're seeing in the urine is a reflection of dietary exposure, not exposure indoors. However, this still may be useful information because it can help us understand what these exposures are, even if they're minor to our overall exposure. It can help us understand exposures that are occurring that are non-dietary. Again, it just depends on your research question. All right, one other thing we get is, do wristband levels correlate with internal dose better than house dust, particularly for chemicals where we know the indoor environment's really important, right? So here's a table I put together, and all this data is actually published. It's just published in 4 different papers. So I pulled it together into this table. So basically you're looking at correlation coefficients between the measurement of the parent compound in one of these compartments, either wristbands, a hand wipe, or dust, relative to its biomarker of exposure, the gold standard. Right, each row is a different chemical. Most of these, they're biomarkers in urine, all of them except for the one on the bottom, this BD47, which is the biomarkers measured in blood. And so if you go across in a row, you can see that the correlation coefficients generally decrease when you go from wristbands to hand wipes to dust, suggesting that the wristband is a better predictor of internal dose compared to, in some cases, hand wipes, but certainly house dust. You'll see for many of these chemicals that the correlation coefficient is pretty similar for hand wipe and dust, or for hand wipe and wristbands. And we've seen that several times. I think suggesting that they both work pretty well, but the problem with the limitation with the hand wipes is that they have to be collected by research staff. So you have to be with the participant, and it's a one and done. So once you analyze it, there's no more left. And actually, when we compare them, the signals are much lower on the hand wipes relative to the signal that you get on wristband in terms of how much is accumulating in those samples. So it's definitely still suggests to me wristbands are a little bit better than hand wipes. But you can see in all these examples, they have a much stronger correlation. And that probably makes sense because it goes everywhere the participant goes, right? As opposed to just measuring what's in one sample of dust. And it's important to note that dust in your living room is going to be very different than dust in your kitchen or in your bedroom because you have different sources. Right. So if your question is what's in your living room, then maybe dust is the appropriate sample to analyze. But if you're interested in what's getting into the body, wristbands might be a little bit better. Okay. Another question we get is, how variable are the levels on the wristbands if you took it from different parts of the wristband, right? So if I took a scissors and I took out a sample here versus here, how similar are they going to be? Is this going to add to any variability in our exposure assessments? So we've done this analysis from 10 participants. This is actually in our firefighter study. And so what I'm showing you here are the results of basically analyzing those 3 different sections and reporting on the average relative standard deviation of those 3 measurements. We restricted our analyses to chemicals that were 100% detected in all 3 wristbands. And so you can see across the board for most of our target analytes here, the values were usually less than 20. THP certainly was a little bit higher in this case, but almost all of them are less than 20, which is ideal. Overall average was 13%. So we do believe that it's pretty homogeneous around the wristband. Okay, how do they compare to spot urine? A lot of epi studies use spot urine. And then are chemicals washed off during showering? These are 2 questions we also receive a lot. So I wanted to really answer that first one. How do they compare to spot urine? Because I was really curious. You know, we've analyzed urine samples for organophosphate flame retardants quite often. And if I take a sample in the morning and take another one in the afternoon or evening, you get very different values because it's a really short half-life, you know, in a matter of hours. And so my thought is that it might be better to use wristband for these chemicals. And I wanted to be able to look at this a little bit more. So in order to do this, though, we needed to collect 24-hour urine samples from participants, which is a big bear, right? So we had 10 volunteers. So this study is certainly limited in our statistical power, but we did have 10 volunteers who collected 24-hour urine samples for 5 continuous days into those big jugs. And then we said, you pick randomly a spot urine sample to set aside on those 5 days. We didn't tell them what time to do it, when to do it, just on each of those 5 days, take a spot urine and set that aside. So we also had a spot urine sample. And then what we did, you can see, was we analyzed the urine. So this is actual data from one of the participants for the flame retardant metabolite TDCPP. You can see on many of the days, it was actually pretty consistent, but day 2 was certainly twice as high as day 1. And at that same time, we asked them to wear wristbands. And we said, when you started off, they started off with 5 wristbands, 3 on one hand and 2 on the other. And each day they would take one off so that we could look at the uptake of these chemicals on the wristband over the 5 days. And so we could plot that concentration. So again, this is actually real data from one of our participants for the parent compound TDCPP. And you can see actually a pretty nice linear uptake of this compound, right? And then that slope reflects that exposure for that individual. But this allows us to kind of make these comparisons, right? Of how well do the wristband measurements here plotted on day 5 So the top row is a flame retardant called TDCPP. The bottom panel is a sister compound, TCPP. The y-axis is total mass excreted over the 5 days. So we summed up all of 5 days. For the panel on the left, those are wristband measurements. So wristband on the x-axis versus total mass excreted. And on the right are the spot urine samples. So we randomly picked a spot urine sample, and then we plotted the spot urine value versus total mass excreted. Now it's 10 people, and these are Spearman correlations, right? Those are rank sum. So they're pretty similar. So basically the message here is they're at least as good or the same as spot urine samples. I would love to do this in a larger study size because when I look visually at the data, I think there's a better spread linearly of the data on the wristbands than you see in the spot urine sample. There's more noise, but if you're doing a rank sum test, which is what a Spearman correlation is, you're getting basically the same correlation. Now, so it seemed to suggest they're at least as good as spot urine. We also wanted to ask a question about chemicals and personal care products because we do get this a lot. Would they accumulate the same way? So we wanted to answer this question, but use the same samples that I just described. So the 24-hour urine samples, spot urines, and wristbands. So I had a student that worked on this as part of her thesis. What I'm showing you here are spaghetti plots of parabens and triclosan in these 10 individuals. So for example, NPB is methylparaben, then you have ethylparaben, propylparaben, triclosan, and butylparaben, right? And so, does my mouse show up? Probably not. What you can see, right? Here's one participant on the top that had really high exposure over all 5 days. Some individuals had very low exposure all 5 days. The thick bold line in the middle is the geometric mean for all 10 participants on those 5 days. The slope actually looks kind of flat, is on a log scale, so it kind of gives that perception. It increases a little bit. Now it's 10 people. If you do a test, that slope is not technically different from 0, but as you go across and the chemicals become bigger and more hydrophobic, the slope is statistically significant. So methylparaben, for example, might at least be at a steady state or slightly increasing, but you can see the variability among the 10 individuals, right? So how does this compare to the urine samples and the total mass excreted? Here's data. This is for methylparaben here, the same kind of a plot. Methylparaben on day 5 of the wristband versus total mass excreted over the 5 days. Correlation coefficient is 0.62, p-value equals 0.05. On the right now, you're looking at spot urine samples. So in this case, we did something a little bit different. This was a great idea by my colleague, Kate Hoffman, right? Because we had 10 people that collected spot urine samples on 5 days. So depending on what spot urine sample you pick, you could get a very different correlation coefficient. So what she did is she wrote an R code to run 1,000 permutations on the different pairings of spot urine samples from the different days and said, what kind of a correlation would you get? And is it statistically significant? So when you run those 1,000 permutations, the correlation coefficients range from 0.15, so not at all correlated, to 0.88, which is strongly correlated. Only 24% of those permutations were statistically significant, and the median was 0.56, right? So to me, this suggests that the wristbands are probably a better predictor than the spot urine samples. This happens to be a chemical that has pretty low ICC when you look at interclass correlation coefficients. They're usually pretty poor, 0.2 to 0.3. So I think in this case, some of the wristbands might be providing a little bit better data on average exposure over time, even when most of these might be coming from personal care products. So this was just accepted for publication, and SCEHSC should be out pretty soon. And then we have the data for the other parabens and triclosan as well. And then here's just examples of other chemicals from that same study and how they accumulated. So not the parabens, not the organophosphates. Here's an example of a phthalate and how these are all individual plots from the different participants. Phenanthrene and transpermethrin, right? So phenanthrene is a PAH, a combustion byproduct. You will notice that the scales are very different for these chemicals, right? Because there's certainly moderate exposure to diethyl phthalate, certainly linear uptakes. Phenanthrene is much lower, but clearly detectable. And then transpermethrin here is on a log scale because some people have very high exposure to this and some people have very low exposure to this. Or again, depending on how you use these pesticides around the home, but still linear positive uptake. So all these people were taking showers every day. That was the point I wanted to make here is like, so I don't think showering is influencing what we're seeing because they still accumulate in a linear fashion, even though participants in the study were showering every day. Because we tell them to wear it all the time, wear it in the shower, wear it to bed, just don't take it off. We don't want you to change your behavior. Right, so just wear it and try to forget you're wearing it. and we still see these uptakes. So that's a little bit about some of the studies we've done to understand strengths and limitations of the wristband. I want to show you some applications where we've used this to address specific questions as part of the research that's ongoing in my lab. One of my favorite projects right now is working with the firefighters to understand their exposures. Many of you may be aware that in 2022, the IARC, International Agency for Research on Cancer, classified the occupation of firefighting as carcinogenic. A lot of this is because data that's come out from the 9/11 disaster and understanding that cancer risks are certainly increased in this profession. So there's a lot of interest in understanding which exposures may be driving this and what can we do to help firefighters mitigate this exposures and reduce that cancer risk. So back in 2019, we started working with firefighters in North Carolina. So this is a project that was led by one of my PhD students, Jessica Lavasser, where we wanted to ask the question, what do your exposures look like off duty versus on duty, but also how do they change when you respond to a fire call versus just being in the fire station? So we started this in 2019, and we just had firefighters wear a different wristband each shift until they documented a fire call, and then the pandemic hit, so we had to stop it. So we only got complete data sets from 20 firefighters, but we actually learned quite a bit from this study, right? So here's an example from one of the analyses for PAHs. Right? These are combustion byproducts. You'd expect them to increase in terms of a fire. So on the bottom are just the different PAHs across from left to right. These are results from a regression model where we looked at the average differences off duty versus on duty. So our reference value here at 1 is their off duty level on average. And what you can see are the circles and the squares are elevated above that. The y-axis is the multiplicative change in their exposure under those different conditions. And you can see that the squares, right, if it's a filled symbol, it's statistically significant. The squares are more elevated than the circles. So their exposures to PAHs, almost all of them, were higher when they are working and responded to a fire as opposed to off duty. What's interesting to me is that they're also elevated compared to off duty, even when they didn't respond to a fire. Why is that, right? So either something's being tracked back or there's a different additional exposures to PAHs around the fire hall or the fire truck that's contributing to these elevated exposures, right? So each firefighter was their own control and we kind of compared back to their off-duty exposures. We saw something kind of similar with the brominated flame retardants. Here you can see again, different brominated flame retardants across the bottom. The ones on the right that say BDEs, those are the polybrominated diphenyl ethers. Those have been phased out now, but we still have exposure to them. But what you see when you look at this comparison is that the circles and squares are very close to each other, showing that they're not really different when they responded to a fire versus not responding to a fire. In case many of these are the circles are higher than the squares. They're not different technically, statistically, but why are those exposures so much higher when they're working compared to off duty? And it doesn't seem to relate to a fire at all. We don't know the answer to this, and this is something I'm really interested in exploring further. Most of the firefighters in the study came from 2 stations in Durham. And I remember looking at this, and I actually have been in their fire station several times, and the firefighters at Station 4 in particular have a very small station where they have a small kitchen, a living area, and then a small table, and then a hall for their bunk rooms where they sleep. And the furniture, I remember, looked really old, right? So many of these BDEs were commonly used as a flame retardant in furniture. And so I asked them, I'm like, oh, where does your furniture come from? And they're like, I don't know, it's a hand-me-down, so-and-so gave to me. It's been in here for a long time. They have no budget to buy furniture for their fire hall, so everything's a donation. So I asked them if they would let me take a sample. Can I open up that cushion, take a sample? And I did, and I brought it back to the lab, and sure enough, there were PBDEs in it. So I can't prove that, but I think some of this is actually from the exposure they just got in the fire hall, just spending time around that furniture, because that's exactly what we've seen in the general population. Now, this doesn't explain everything, because if you look on the left, DBDPE, for example, all the way to the left, that's a replacement for the PBDEs. We know it's heavily used in a lot of applications. It's a high volume production chemical. I just don't know where that exposure is coming from. And I think that's also very important to look at further. Right. But then we even look at something like phthalates. Now, phthalates didn't really show a big difference off duty versus on duty. And if anything, some of their exposures were higher off duty compared to on duty. Right. And these are the ones that we know are often used in personal care products. So I think this just reflects differences in their personal care products used when they're working or versus not working. We saw the same thing for several pesticides. right? And so Nick is really interested in this data, has been taking it, doing some analyses with suspect screening. So I'm just going to show you some volcano plots. I'm sure most people are familiar with them, but each dot represents a feature that was present in the sample. And we're kind of comparing them in two different conditions. So I'm going to show you data versus when they're on duty responding to a fire versus off duty. And all of these analyses are restricted to features that were detected in at least 50% of the samples and were already blank subtracted. These are all non-parametric analyses. So I thought this was very interesting when we looked at this, right? Obviously, we want to identify what these chemicals are, but I think it's also important to look at the patterns that we see in some of these, right? Getting back to that chemical fingerprint. What you can clearly see if you have that lined in the middle is that there's non-symmetry here. There are more features that are showing up in firefighters when they're off duty compared to on duty, which I thought was interesting. I might have expected the opposite. Why is that? I don't know. Maybe there's just more things we're exposed to in our home environment. Now, we did have the target analytes, right? And so what we were really interested in are these features. They're in the upper right-hand quadrant. Many of those were things that were in our targeted panel, like a lot of combustion byproducts that make sense, PAHs, some flame retardants. The one that was interesting to me is this one is 6,2-FTMAC. This is a PFAS. It is one of the PFAS that is known to be in the outer shell of the turnout gear. So it is possible that this is coming from exposure from the gear. I don't know if it is or not, but it's something we want to follow up on. And this one is now in our panel as well. Right, what we did, that was our first study. We already did a second study just very recently. So this is getting to my point of looking at long-term exposures with wristbands. So we had, in this case, 29 firefighters volunteer. They provided a blood sample when they consented. And then we gave them a bunch of these wristband kits and asked them to put on a new wristband for every work shift period. In this case, firefighters have this 9-day period where they're working 24 hours on, 24 hours off. So it's 5 working days, but then they have 4 days in between when they're not working. But to make it easier for them, we just said, just keep it on that 9 days. And so they did this for a period of 4 to 5 months. They came back, they returned the wristbands, and we got a second blood sample. So we're looking at the blood for PFAS and for these PAH adducts, because I'm really curious to see how exposures over time relate to PAH adducts in the blood. So those analyses are ongoing. I have some preliminary data here I was just going to share with you. Right, and this is just an example of what this looks like, right? Each color represents a different wristband. So most firefighters had 6 to 8 wristbands they wore during this period. So we could look at those exposures over time. What was really interesting to me, I'm interested in these flame retardants. Here again are just spaghetti plots. These are just from the first 10 participants in the study, but the ICC values are from the entire set of data. So you can clearly see that the flame retardant decaethane on the right, a little bit higher exposure in general, but very consistent exposure over time, which I found very fascinating. Whereas BD209 on the left had lower exposure, but really variable. Why is this? This is what we're trying to tease apart because we do have their fire call data to see if some of these ups and downs are related to fire calls. I have a feeling that the decaethane data now suggests something in the station itself is leading to exposure if it's that consistent over time. But we need to take a look at this a little bit more. Here are these kind of plots that we're using to visualize the data right now. So each panel represents a different firefighter. The dot is their wristband measurement. Red is from when they wore it and they responded to a fire. Blue, they were wearing the wristband and didn't respond to a fire. But you can see there's definitely a spread in the data, but some individuals have really consistently high exposures, and others have low exposures. We don't know what's driving this. Now, if you just do a simple analysis of whether there's a difference by fire call or not, they are elevated statistically higher in fire calls versus not fire calls, but they're really consistent within a firefighter still. And so I'm really curious to try to figure this out. We have just started analyzing their gear. You know, there's a lot of interest in PFAS in gear. We got samples of moisture barriers. So that's there's 3 pieces to turnout gear. The middle piece is a moisture barrier, which is typically Teflon or PTFE for the water repellency. But we are finding decaethane in some of the new moisture barriers. Now, whether that's where this is coming from or not, I don't know. And I don't know how common it is in all moisture barriers and all turnout gears. We've analyzed 2 samples so far, but this is something we want to follow up and see if that's where this exposure is coming from. All right, so I'm going to pivot and just really talk quickly about our One Health study. This is being led by a postdoc in my group, Kathy Wise. We're interested in understanding, I've heard of canary in the coal mine. Kathy likes to say you have a canine on the couch to understand how, if our pets can inform us about exposures that are related to health. Right, so there's an interest obviously in One Health studies, and we wanted to ask the question of whether we could use these wristbands on our pets as well to understand exposures. We first had to start with the basic questions. Do we see a correlation with internal dose like we see them in people? So here you can see one of our participants, they have on their collar, a little keychain with a little piece of a silicone wristband. So we just cut up pieces from wristbands and we put them on the collar here, and the dog wears them for 5 days. Then we also have the owners collect urine as well from the dogs. So we focused a lot on pesticides, and I'll explain why here shortly. So what you're seeing here are correlations again between wristbands and urine, on the top for DEET and the bottom for permethrin. These again are pooled urine samples. We had the owners wear a wristband as well and collect their own urine, so we could look at this comparison between the pet owner and the pet itself. So you can see on the top, DEET is nicely correlated, again, between the dog and between the owner, between the silicone tags and the urine. And we see the same thing for permethrin on the bottom. They're all statistically significant. What's interesting to us is we're seeing this trend that the correlations are actually tighter in the animals than they are the people. And I think that's because the animals have more routine behavior every day than a person does. Well, then they said, well, are we seeing very similar exposures in the pet owners versus the pets? And that's also a yes. Here are 3 different chemicals, different classes. Where here I'm just comparing what accumulated on the dog's tag versus the owner's tag. So even though the dog's probably home, the owner's going to work, we still see very similar exposures. For a legacy PCB 28, right? PCBs are old pops. We still detect these, they're correlated. A new flame retardant, this is a flame retardant in what's called Firemaster 550, that was highly correlated. And then there's that DEET again, very nicely correlated between the two. We also had a survey in this case, and we asked the pet owners, what flea and tick medication are you using? So they told us exactly which one they use. And so we categorize this based on the active ingredient. So some of these contain fipronil, right, which is an active pesticide in these flea and tick medications. 9 of our 30 participants said yes, they were using one of these that had fipronil. And if you just split the samples up based on yes or no, if their owner reported using it, you clearly see a difference in the levels that are being measured. So on the left are the dogs, where there was a 100-fold increase on average in the dogs being where they had a flea and tick measurement with application with fipronil versus dogs that did not. And then we saw the same thing in the owners, right? So on the right are the pet owners. We don't know if they were always the one applying this, or if they applied it while they wore it. Probably not, because there was only 1 week. But you can still see a 10-fold difference between the pet owners who reported having fipronil on their dog versus those that did not. So the point of doing this is because we wanted to support a study on bladder cancer in dogs. Kathy's advisor, Matthew Breen, is a veterinary oncologist at NC State. His group has developed this really nice bioassay in urine for measuring this BRAF mutation. So this can be detected in the urine that the cells shed in the urine. And if you detect it, even when the dog has no clinical symptoms, their data suggests that if you follow them up for 2 years, there's an 80% probability that that dog will develop bladder cancer. So we wanted to understand if we could look at like gene environment interactions here. So they have used resources available that they had through the AKC to advertise this study. And so people were mailed kits where they collected urine and they mailed it back, and it was tested for this mutation. If they had the mutation that was less than 5% fractional abundance, they were a case. And then we found breed matched and age controls in a 3-to-1 ratio, 75 controls to 25 cases for this BRAF mutation to look at environmental exposures that may be related to risk for bladder cancer. And we did both targeted and non-targeted analyses on these samples. The targeted data didn't provide a lot of data. We actually saw a few flame retardants that were statistically higher in cases relative controls, but it wasn't a huge difference. But I'm going to show you the non-targeted data here. This is just quickly where everyone was located. This is across the country. The orange are the cases, the blues are the controls, right? We matched on breeds, the type of breeds in the study and the dog age and the sex to control for that. And we specifically asked about tobacco use in the home because that is a risk factor for bladder cancer. So everybody basically said no, with a few exceptions in the controls. But here's that volcano plot of cases versus control. So again, you see this more features on the right. If you look at this pattern that emerges, suggesting there's more exposure in our cases relative to controls. And again, we have a 3-to-1 ratio of controls to cases here. So you can see where these fall relative to the p-value. I did not correct for multiple comparisons here, right? We kind of think about this as really hypothesis generation. So we're really interested in trying to identify these features in the upper right-hand quadrant that are elevated in our cases relative to our controls. We're still kind of processing this. The data does suggest that it's industrial chemicals and pesticides, but that's not everything over there. And obviously this needs to be repeated in other studies. But because this was a national study, we collected a lot of metadata that we could look at, again, for these patterns, right? So we looked at differences by latitude. Where did the dog live? You can see if we just split them at the median, there's more features on lower latitudes than higher latitudes. You don't see that same discrimination when you look at it by longitude, which made me think that temperature was playing a role here. So if you just plot it based on the average outdoor temperature from where the dog lived when they wore it, you can see many more features on the, when you just split it again at the median, what the temperature was on the higher temperatures versus lower temperatures, which I think is kind of fascinating, suggesting again that just where we live is playing a role in what type of exposures we receive. In-home pesticides, did they report this? Yes versus no. You can clearly see that when they said yes, there's more features being detected. Lawn pesticides, same thing. Some of these are pesticides, they're not all pesticides. I think this is really more of a reflection of the home environment, maybe conditions that vary based on socioeconomic status, would be my guess. We looked at just differences by sex of the dogs where we didn't really see something, which probably makes sense, but we did see something in the firefighters that was interesting to us. If you look at the plots here, there were more features that were statistically significant in female firefighters versus male. We only had 5 versus 15, so it's small, but Nick looked at this in 3 other cohorts and we see the same thing. The top right is a cohort from North Carolina where we had more females than males. The bottom 2 are actually samples that are collected through a collaboration with Harvard, where we looked at levels in the US versus the levels in China. Oh, I didn't even see the power thing come up. Thank you. It's okay. Oh, there's one on here. Thank you. So we saw this difference in terms of the features that were identified in female participants versus male participants. The bottom 2 are US office workers on the bottom left. The bottom right are office workers in China, right? So there, if you're going to look at the upper right-hand quadrant, you can see how many of these features were statistically significant and a p-value of 0.01. We're not correcting for multiple comparisons. But again, I'm interested in these patterns that are emerging. So it does seem to suggest that this overall females have more exposures than males, which likely seems to be driven in this case by personal care products, because a lot of the features we have identified so far are chemicals that are associated with personal care products. What was interesting is that our collaborators from Harvard took this a step further. Here are these 2 publications they have, if you want to look into this further. The first one was, again, just looking at office workers in the US versus UK, India, and China. And again, the differences in exposures were really striking between the different countries. Clear differences in flame retardants, which reflects different use of flame retardants in different countries, pesticides, and then India and China, we saw higher combustion byproducts than we did in the US and the UK studies. They were quite statistically significant. But then Anna Young, who was a postdoc working on this, was really interested in whether the chemicals accumulating on the wristband could be used in a toxicology study. So she worked with a group where she extracted everything off the wristbands, and then they ran them in cell culture to look at thyroid hormone receptor antagonism, androgen receptor antagonism, and estrogen receptor agonism. And the biggest difference they saw was between males and females, whether that wristband came from a male or female participant. So here you can see the data for those 3 different cell culture assays. And there were some slight differences between countries, but the biggest difference overall was whether that wristband was worn by a male or a female, right? It was interesting that the thyroid activity data was higher in males versus females, but the other 2, it was higher in females compared to males, and quite a big difference here on the estrogen receptor antagonism between, again, chemical mixtures on the wristband and whether that wristband originated with a male or a female. And then they ran some advanced statistical models, which I'm not as familiar with. You probably have more familiarity with these. This is a Bayesian kernel machine regression model, where they looked at the impact of the influence of the mixture in these wristbands and saw statistically significant association over these different quantiles, right? So it's suggesting again that these mixtures that we're exposed to are different based on whether you're male or female, and also maybe contributing to some of these measurements of toxicity in these cell culture assays. What this means biologically, I think, is still yet to be determined, but it just gives you a sense of how you can use these in other applications. So anyway, I know I've been talking a lot. I am going to stop now. I know I'm biased. I'm a big advocate for using these, but I think they can help us answer lots of questions. They're not invasive. They can be mailed back and forth so that we can reach populations that are hard to access, that you can't get in and get blood and urine samples from. You can mail them. We mail these out, and we have a YouTube video that people can watch to show how they should wear the wristband and how they should return it. And I think these can help us a lot understand of how are these exposures over time? How are they occurring? I'd love to do one where we kind of look at differences in winter versus summer or fall 4 times over the year to see how it changes, how it varies more geographically. So I think there's a lot of potential here, right? We know that a lot of our exposure, at least what we're measuring with these wristbands, is driven by behavior and the built environment. We really need to understand these exposure patterns a little bit better, particularly in the light of climate change, increasing heat. There's a lot of changes in consumer preferences over time. And I would love one day to see us be able to use these and have them integrated with like a GPS tracker, right? An accelerometer, so we know like how fast we're moving. I think there's a lot of potential there. And so I'm hoping we'll reach that point maybe in my lifetime, and I understand all the data that we can collect. So I just want to thank my group. There's a lot of people that always contributed to this, and certainly acknowledge all the different collaborators and funding sources and colleagues and friends and industries that help support our research. So I will stop there, and I'm happy to answer any questions if there are questions.
Thank you so much, David, for this wonderful discussion. Now, we have some time, but opening to questions.
And there's one right over here. Go ahead.
So this is really fascinating, and this is my colleague from Ulaanbaatar, Mongolia,
Mhm.
and we study pollution in the world's worst polluted city. So one question is, how much pollution can you take as an asymptote of these things?
Yeah, so that's something I don't think we know the answer to, right? You're asking if they reach a saturation
Well,
point.
Yeah, I mean, in the winter there, it gets up to a gram per cubic meter.
Mhm.
Is that too much for your wristband, or is that?
Well, that's air, so I don't know how that would translate to a wristband, but keep in mind that this is time dependent and where you spend your time. Most people are not probably spending time outdoors, right? They're indoors.
They have to commute to work, and they go massive photos when they commute to work.
Mhm.
And indoors, it's very polluted as well.
Mhm. Right.
But anyway, so aside from that, anyway, I think it's definitely fascinating, and this will be, the reason we're here is the sort of title of your talk, and we'd like to invite you to collaborate with us on doing a study in Ulaanbaatar using this, because having personal exposure, having something simple like this is really, really, really cool.
I don't disagree. I think they're super cool. I'm happy to chat with you about that later. Yeah.
Well,
Our issue is bandwidth right now, so.
Well, I invite you to come to UV and sort of scope it out. I'll be going in June and July. If you want.
Thank you. Well, we can talk.
Thank you.
Any other questions?
Taylor?
Are there a lot of labs that can analyze the data, or is it just your lab that can do this?
Yeah, it's not just my lab. Kim Anderson's group at Oregon State is also doing analyses with wristbands. Actually, her former student now actually runs a company called Exposome, where they offer these commercially. It's just important to note that they're just set up a little bit differently than ours. So they're not doing the non-targeted analysis part, right? And a lot of what you measure is depending on what type of analytical instrumentation you have. So that's a little bit different as well. But there are more researchers interested in trying to bring these online in their labs. I know Doug Walker at Emory University is using this. Marta Venier at Indiana University is also using them. And then Crystal Pollitt at Yale University has something similar. It's not a wristband, it's called a fresh air band. Looks a lot like a smartwatch, and it has a compartment that you open, and inside is a PDMS cylinder, right? So that has advantages because it can pick up gas phase very well, and you don't get those skin oils. So she can take that and just basically drop it into her inlet and use thermal desorption to measure those. The downside is that you don't capture those particle bound chemicals in this design. So it works better for gas phase than it does particulate phase, whereas the wristband, we're capturing more of them, but we do have more time then to clean up the extracts before we analyze them. But it's also a very nice wearable sampler. Yeah, but there's definitely interest growing. Yeah. Lydia?
Yes, my question is more technical about PFAS. So what is the correlation between, PFAS measurement in wristbands versus, house dust versus, hand wipes?
We don't have hand wipes in any study where we have dust and wristbands. We do have these two studies now. One is in California, one's in Michigan. Actually, we're doing something similar with firefighters where we have paired samples of blood, wristbands, passive air. So we use what's called a SIP air sampler, passive air sampler for 3 weeks. And then we have the dust collected. So we've really analyzed the Michigan cohort data first because the California is still ongoing and being collected. PFOA and PFAS, we do detect them in house tests, certainly. Some homes have quite a bit, at least in Michigan. It's not correlated at all with the blood. And I just think that's because most of the exposure, at least in that cohort, is from drinking water exposure and food exposure. So that happens to be a community where they had a paper plant that was using PFAS, and it got into the waste that was put in a landfill, and then it contaminated the groundwater supply. We need to really do this in areas where there's no PFAS in the drinking water to better understand these relationships between the indoor environment and these ambient measure and these blood values. But I'd also love to see us look at other PFAS outside PFAAs because everyone's doing the same panel. And what we pick up in the indoor environment are these precursors. Yeah, that theoretically can go down to PFAAs, but I imagine there's a lot of these intermediates. There's conjugation. There are probably better biomarkers we could look for in blood for these precursors. And I think that's a big gap right now. So did that answer your question?
Yes, in regard to the correlation between dust and, house dust and wristbands in the precursor of PFAS?
Oh, I have to go back and look at that data. I've really been focusing on how well they correlate with blood. We did see very nice agreement between the passive air samplers and the wristbands for many of the compounds. I think a few were correlated between dust and wristbands, but not many of them, and not for the ionic PFAAs, like the carboxylates and sulfonates. Sure. Rima?
Thanks so much for this. So, I did a lot of personal monitoring for air pollution particles and gases, right? And we think a lot about the personal cloud that means like this ghost that's following around. But I've only thought through it through like a VOC, for example, like if you're doing personal monitoring for VOCs, potentially your breath fixation in your skin. Are there similar examples in the setting that you're thinking about sort of things that our bodies are emitting that affect some of it?
That's a good question. I think I haven't thought about it in that detail. We do get the question a lot about whether what we pick up on the wristband is what we sweat out basically from the body. I don't think that's occurring. I have limited data to support this, but in one case, what we did with 3 people is we took a wristband, cut it in half and layered it and put foil in between. And we had people wear that for 3 or 4 days. So the bottom one was in contact with the skin and the top one was not. The values look the same, basically on both of them, at least for the flame retardants. We didn't do the whole panel, which we probably should have. So I don't think for many chemicals, it's reverse causation or what's being sweat out. I do think you're going to get differences based on whether, I know some groups will put a passive sample like on a brooch. I think it was Crystal's group that did the comparison between a wristband, a brooch, and on your ankle. And they certainly they were correlated, but you had more signal on the ankle, and they thought it was because they were kicking up the dust, so you got more particles on there. You are going to get differences depending on where you wear it. Yes. One of the other fascinating things we learned that I didn't put in here, but in our PFAS cohort in Michigan, when we detected these precursors, for example, we measured 6,2-dipap pretty commonly on the wristbands. That's a PFAS that we know is in makeup and cosmetics. And we had asked the participants, do you use mascara, do you use foundation? And when you look at that data, there is a trend. We saw higher levels on the wristbands if they said, yes, they use this makeup. Now, we can't confirm that their makeup had it in there, but it makes me wonder if, these are off-gassing maybe from in the personal cloud and our wristbands pick it up. I don't know. It needs more attention, but it was really interesting finding, I thought.
What you kind of being done in children, in adults, or people done in children?
No, some of the slides I show were from our toddler cohort, which we call TESSI, which were 3 to 6 year olds. And we're now actually piloting an anklet for infants, all right? Because we really want to be able to get younger children too. We don't want it to be choking hazards. We don't want it on their wrist. So we've designed these very small ones. They're a little thicker that goes on the anklet. So they're actually being deployed right now. We're getting urine from the infants. So we'd like to see if they work at capturing exposure so we can target even younger children than 3 years old. Yeah. Yes.
So the cost of this is in the analysis, and the cost of the analysis depends on what you're looking for.
Mhm.
And how well, you said they're pretty stable if you freeze them. Could you count on many chemicals being stable over a really long period? Like, getting the wristbands really cheap. You can do that on a whole cohort potentially, and then you could archive it and.
Yeah. And that's what.
Pull it out a lot, you could do it on like 1 million people probably.
Right.
The cost of the wristband. Is that something you've thought about?
We are doing everything we can to scale up because, so my lab is part of the NIH HERE program, Human Health Environmental Analysis Resource. HERE is also supporting some of these large cohort initiatives. I am was supporting part of one of an echo cohort, but that's kind of finished now because I think that was not renewed. But there are these seeker cohorts, which is cancer in the environment. So we are supporting 2 of these cancer cohorts from NCI with wristbands where they're recruiting thousands of people. We're supporting a pregnancy cohort at Duke. We're now supporting through the HERE program this CURE initiative, which looks at chronic kidney disease in different countries. So we're supporting wristbands that are being distributed to these different areas of the world. And now we're also helping the National Firefighter Cancer Cohort Study, which is deploying wristbands as well. So we're trying our best to scale up and support all these studies. And everyone's just archiving them for now with the hope that we can analyze them later. Some of the data I showed you today was in our freezer for 2 years before we had the bandwidth to analyze them, and we still saw associations with the urine. So I do think they're fine for at least for the classical SVOCs that we focus on. Now, we don't look at a lot of volatile chemicals in my lab. Those could be very different. And we don't look at metals or other things, right? But I have a feeling, or my gut tells me based on our data, that they're pretty stable in a freezer long-term for years. How long, we don't know. That hasn't been assessed.
Bandwidth, no pun intended, isn't it?
Pardon?
Bandwidth, no pun intended.
Yes. That's our issue, right? We're trying to automate as much as we can. We just purchased a custom oven to help us clean like 200 wristbands in a day, as opposed to the 50 we can do in a day right now. So scale up is certainly the factor.
The unit cost for the analysis.
It does depend on whether you want targeted or non-targeted and which panel. Our basic one is, and this is just, most of it's labor costs. It's all analysis, right? They're cheap to buy. To break even for us, because we use high resolution mass spectrometry, which are expensive interests, it's $250 a sample for us. And if you go all the way up to non-targeted analysis, we get estimates around 600 a sample. So it's not cheap, and I get that. I'm hoping in the future it gets there. Yeah. I'm hoping in the future this gets a little bit easier. We can automate it, find easier ways to do this. Particularly with the non-targeted data, we have these algorithms, but we still need to work on improving them for accuracy of feature identification. That's going to take some time. But as I said, I'm interested in these patterns. These patterns I think are really interesting, and we can learn a lot from them, even if we don't know exactly what those chemicals are.
Do the commercial operations, do they get, you mentioned a company in Oregon, do they get economies of scale that, you have something specific that you need to do?
I don't know exactly.
You send somebody to them.
I think they were charging $1,000 a sample, yeah. Mhm.
For the exposomes, no.
Mhm.
But for targeted, it should be like cheaper, no?
They're only doing targeted. They don't do non-targeted.
All their money.
At least that's what I know. I am supposed to have a meeting with them next month, so I guess we'll see what else they're doing. I don't know. You had another question in the back? Yeah.
Did you ever came across like false positive detection or is it purely like a feature annotation?
Certainly false positives in the non-targeted analysis, right? Because each feature on there, we have a list of tentative IDs, that can go down to 30 possible identifications, and we rank them based on metadata we have, so that we like to assume the top one is probably what it is. But we found a few cases where it was not, and it was number 3 on the list, right? So it's certainly very challenging, and that's where I think we definitely need more work. There's a lot of effort going into this, different algorithms, different approaches, and an effort to improve the mass spectral libraries, which are going to be important. But some of these could be transformation products, right? So they're not going to be a pure chemical that's in your database necessarily. Yeah.
One final question, what's the influence of these concentrations that you found in wristbands? In other words, how these results could be applied to policy regulations?
Mhm. Right.
How can we interpret this? What does it mean?
Yeah, no, that's a great question. We do get that as well. You know, I again have this vision where in the future, we can use AI and machine learning approaches to take a wristband measurement and it'll tell something of what it means about the levels in the blood.
Oh, the prediction model.
Yes, yeah, that's where I want to go. Now, other groups are saying, well, you can take what's on there and then you can, using some calibration approaches, estimate what the air concentration was, and that can give you an inhalation route. My concern with doing that is all the studies that have done that to date are in chamber studies where the samplers are static. We move around, we're active. And we have a study under review, and I didn't get to talk about it, but we have shown that movement increases uptake rates, right? So if it's stationary versus someone just at a walking speed, you can have a very different slope. Your uptake rate's very different. So I want to use caution with taking what's on a measurement and inferring an air concentration. Plus, some of these are integrating inhalation and dermal, and that's the fine line. We don't know where inhalation stops and dermal starts, which to me is why I like the wristbands, because maybe it doesn't matter. We need to use these approaches to understand what they mean for the chemical levels in our body. And that's where I'm hoping we can get to the policy risk. And I just think, I personally don't want to see us use them for measuring air concentrations when back. But that's my bias there.
Thank you.
Yeah, Rima?
Just a quick thought, it's fascinating, right? And probably by placing them like in key areas, let's say like very high up in the room versus close to the ground, plus the person, right, inform a lot of kind of, flow models and exposure models and pathway analysis too. It's really helpful.
I will say I've done that in my own home where I put them out for 28 days and then I had myself wear them and my husband wear them and my kids. At 28 days, for many of these chemicals, it reaches maybe 20% the levels that we get on us wearing them for 5 days. Like you just get much greater accumulation over time when you wear them versus when they're stationary. And to me, that's the movement issue.
Okay.
And the personal cloud. Yeah, exactly. I think that's a contributor too. Yes.
So
Yeah.
one of the great advantages of doing this in Ulaanbaatar is that there's this very marked seasonal fluctuation in pollution. We call that cold in winter. And we already know that there's a very strong dose response relationship between air levels and various health outcomes. So that would be kind of cool. Now we can get down to what components might be doing it.
We don't typically measure PM 2.5 on these. That's just not what we do. Right. Yeah.
Say some more about this dermal respiratory issue. And that some of these chemicals are well absorbed thermally and some of them are, there's no dermal absorption.
Right.
And I mean, that must affect how well it, if you have a lot of dermal exposure to a chemical that's a respiratory toxin, it's in, the route of exposure is through inhalation. It's not gonna work very well. At least it's not gonna work very well if you have dermal exposure.
Well, again, but they're integrated. They are measuring what's in the air, but they're also picking up, because right, our skin is a passive sampler on its own, right? And if you look at some of the work done by like Weschler and Nazaroff, they show that, chemicals in which maybe we thought initially inhalation was the main route of exposure, some data is now pointing to the fact that these chemicals condense on our skin, and even though absorption is very slow, over time, the chronic nature of it leads to significant accumulation in the skin. And they see these trends based on phys-chem properties. But again, defining where it stops being inhalation versus dermal is very hard to do. I think it's somewhere in the middle.
Yeah, but once it gets in the bloodstream, it all goes to the lung.
Sure.
Just from the outside of the alveoli anyway.
Although some of them might be metabolized in the skin a little. I know we have esterases in our skin and you could get some transformation, but yeah. I think it depends right on the loading rates, right? And when it's washed off and there's a lot of factors. So there's still a lot that we don't know about these, but I'm hoping we can contribute to that knowledge and use them in the best way we can to support these questions.
Okay, they just informed me that there is a class here starting at 1, so we need to leave the room, but thank you so much for there.
Yeah,
This
thank
was a
you.
great discussion and a great.